Hi. So today I'm going to talk about proteomics and the specific issues in informatics that we have with proteomics. Oh. Okay. Yeah. So proteomics, and when I talk about proteomics, it's mainly mass spectrometry-based proteomics. So there are also protein arrays that some people work with, but those will be covered by Stuart in the next lecture, and that's because those are very similar to gene expression arrays. So if you go to the website, there are a few files there that you can start downloading already now. There are I think 3 Python scripts and two MGF files with an extension MGF and 1 text file. So we'll use those to exemplify different things. So if you start downloading them now, it's probably a good idea. So what I will talk about is first give an overview of the structure of mass spectrometry data, and then talk about protein identification and then protein quantitation. And the different examples that we'll go through will be related to the protein identification and quantitation. So on the general overview of how one uses mass spectrometry to identify and quantify proteins is that one takes samples, goes through many steps of processing, and then finally ends up with small peptides, and then analyzes these in a mass spectrometer. And there are 2 things that are measured in mass spectrometry. One is the mass to charge ratio, and this can be measured very accurately and will give you the identity of the peptides. And then the intensity, which is not measured as accurately as the mass to charge ratio, will give the quantity. So if we look at the steps to prepare a sample for mass spectrometry, we start out with the tissue or cell culture, and then first you have to lyse the cells and extract the proteins. And here there can be quite large losses. So if you from a tumor tissue, for example, from a biobank, the yield of proteins could be as low as 1% or sometimes even less. But usually with cell culture we can get quite good yields. So then now we have a complex mixture of proteins. So we need to separate to be able to analyze. But we don't need to separate them perfectly, but still we need to go from down to at least not too many proteins that are measured at the same time. So we can fractionate on the protein level, and then what most people do is they digest the proteins with an enzyme, usually using trypsin to get peptides. And then these peptides go into the mass spectrometer. So the general overview of mass spectrometry is made up of 3 parts: an ion source, a mass analyzer, and a detector. And then out of the detector we get an electric signal that is the mass spectrum. So generally in a mass spectrometer, the way we measure mass is that we separate the charged ions in electrical and magnetic fields. So you probably remember that the force on a charged particle or a force in general can be written as the mass times the acceleration, which is, and the acceleration is the time derivative of the velocity. And in an electromagnetic field, we have the force is written as the charge times the size of the electric field times a vector product between the velocity and the magnetic strength of the magnetic field. But this might seem complicated, but the main thing is that we have on one side the mass and the charge, and we can rewrite this so that the mass and the charge is can be calculated independent of just from the strengths of the electrical and magnetic fields. So the meaning is that we can never measure mass. We can just measure mass divided by charge. And that's the general thing in all the... we can, as you see later, infer charge in different ways. But really what's measured is mass divided by charge. So if we then have a look at a few example data. So one ion source that you can use is that you embed your proteins and peptides in a matrix, and then you shine laser light on it and ionize the matrix and your peptides, and they go into the gas phase. And then you accelerate them in an electric field and have a measure how long it takes them to fly down a tube that's maybe 1 or 2 meters. So this is a very simple mass spectrum. So we separate now, we give them all the same energy in the electrical field, and then depending on their mass to charge, the ones that the heavy ions will fly slower and the light ones faster. So this is the kind of spectrum we get first. So if we zoom in to one region, we can see peaks. And what we see is actually, so each of these groups of peaks correspond to one peptide. And we'll look at that a little bit later. But even then, you can zoom in even further, and you see that you have these nice isotope distributions in this case. We can also zoom into an area that has seemingly very little, but it actually turns out that even here there are peaks that correspond to peptides. And but here we see another phenomenon, and that in these spectra we get a peak at every mass, pretty much, because we have such a complex mixture of low abundant peptides that so we need to decide what's a real peptide and what's just a mixture of other peaks. So then we can add in an extra step, which is the fragmentation step. So now we've looked at data where we only measure peptide masses, but if we first have the ion source, we have one mass analyzer where we select peptides, and then we can fragment them and separate them out in the second mass analyzer to get a fragment mass spectrum. And the way that we get the fragmentation is along the backbone of the peptide, and usually at low energy what we get is what's called B and Y ions, which correspond to fragmentation at the peptide bond. So that's and so if we look at some of these, and then we get depending on where the charge is retained. If we have a charge on the N terminus, then we get the B ion, and if it's on the C terminus, we get the Y ion. So then we have another step. Since we've taken a complex mixture of proteins, digested them with an enzyme, and made it even more complex with millions of different peptides, what we do is we separate them in time. So we do chromatography. So at each time point, there's only a limited number of peptides that go into the mass spectrometer. And then we try to collect data as fast as possible to be able to both get survey spectra to get the peptide masses that come at a certain time, and then fragment as many of them as possible to get their fragment spectra. What you will see later in the exercise, there are the two data files. They contain a lot of spectra. So it's easily, one can in maybe a 2-hour data collection collect 50,000 fragment spectra. So there are different ways to show this. This now represents a LC-MS run where we have time on the x-axis here. Then we have mass to charge on the y-axis. And then we see that we have a lot of these spots that correspond to peptides. And in general, a peptide will start appearing at some time and then go away after a little while. But as you see, this is the ideal case, but sometimes there are these streaks. So it's a peptide that appears, but then there's a very long tail, which one should definitely avoid if possible. But chromatography is usually quite hard to do properly. So then if we zoom into one of these, we see that here we have, so the peptide appears and then disappears, and then there are this pattern of the isotope. So this is the isotope distribution that we'll look at closer later. And then at these x's is where the mass spectrometer has decided that it's going to collect and fragment that peak. So then from there, for each of those, we get a fragment mass spectrum. So as you see, there are several dimensions to this. And so we have the time, we have the peptide mass, and we have the peptide fragment mass. And we measure intensities in this space. Yeah. And yes, so the determinant, so it first the instrument takes a survey spectrum, and then it does a quick peak finding. And usually you can, the person that runs the instrument can decide in advance how it wants the mass spectrometer to do it. But usually one would take the most intense peak, isolate it, and fragment it. So that would be and then the next one, and so on. So now typically one would do one survey scan and then fragment each of the 10 most intense ions. It's done fast, so the algorithm is fast, so it's not very precise. So if you run the same sample twice, it's not going to be, you're going to identify slightly different peptides. It's going to, the most intense ones are going to be the same, but the lower intensity ones are definitely going to be different. So then we have the charge distribution. So there are two main ways to ionize. One is the matrix, this laser distortion I mentioned before. The other one is electrospray. And if you have a peptide, usually in MALDI you get the 1+ is the most dominant. But in electrospray, it's usually more the 2 or 3+, and you get more of a distribution. And then when you go up to larger proteins, you get more charges, and in electrospray, many more charges. So you get this distribution. And what we can do is if we want to calculate what the mass to charge is, there is a simple formula. You take the molecular mass of the peptide or protein, you add the mass of hydrogen, the number of times that you have charge, and divide by the charge. So that's how you get. So these charges are protons that are attached to the peptide. Yeah.
You said you divide by the charge, or you divide by the number of charges?
Number of charges. Yeah. Sorry. Yes. Number of charges. So n is the number of charges. Yeah, so in MALDI, what you do is you you crystallize a small molecule. Usually things like nicotinic acid, synapinic acid, and they form a crystal. But you have a mixture of your proteins and peptides. So the peptides and proteins get embedded into this crystal, and then you shine a laser on it, and the laser deposits lots of energy. And the matrix molecules are chosen so that they absorb the laser light at that wavelength. And then what happens is that they explode. In this explosion, they then drag with them the peptides and proteins that are embedded in the crystal and throw them out into the gas phase. And in this process, you get some neutral molecules, some of them charged, and so there will be. It's a complicated, of course, happened this explosion. But in this process, you get some charges attaching. And in electrospray is a much gentler process. So there you go from liquid to gas phase. And so you have your proteins or peptides in a solution. And then they go from the chromatography usually into a needle, and that's at charge. And then the mass spectrometry is at ground potential, and small droplets charged droplets form at the tip of this charged needle. And then when these droplets go into the mass spectrometer, it goes from atmospheric pressure to vacuum. You start losing, the droplets start evaporating, and at some point, since they are charged, they become too small to hold the charge, and then they fission, and then each of these will continue evaporating, and eventually when you're inside the mass spectrometer, you have their naked proteins and peptides with a few charges left on them, without the solvent. So these are the two most common methods that are used. Yeah, so this is just an example. So if we have a peptide with mass 898 that has one charge, then m over z is 899. So we just add 1 divided by 1. And of course, we should, if we do high accuracy measurement, we shouldn't use integers, but we should use the exact mass of the proton and the exact mass of the peptide. But this is just an example. So then the same peptide, if it has 2 plus, it's the m over z is 450. And with 3 charges, 300.3. So that's quite straightforward to calculate. So then we have the isotope distribution, as I mentioned before. So we don't only have carbon-12, nitrogen-14, and hydrogen in nature, but we have 1% carbon-13, and of the other higher isotopes, we have also a little bit. And so what happens is that as your peptide becomes larger, there is a larger chance that you have a few higher isotopes. So for example, that's what we see here is that if we have mass around 1,000 Dalton, the carbon-12 peak dominates. And so this is carbon-12, this is one higher isotope, another higher one. But then if we go close to 1900, the 2 first peaks are about the same in isotope distribution. And then at higher masses, the carbon-12 peak starts to become smaller. So here are the different natural occurring isotope higher isotopes that we can. So we can of course calculate this exactly, but just to this formula changed. So anyway, it doesn't matter. If we just look at the carbon-12 and 13, it's a simple binomial distribution that would give us what the ratios are between carbon-12 and carbon-13. So now, since we have, what I said before is that you measure mass to charge. But if you have an isotope cluster, then we can easily determine what the charge state is. So if there is one m/z unit between the peaks, then the charge is 1. If it's 0.5, it's 2. And 0.33, it's 3 plus. So now, who can tell me what the charge states of these two are? Anyone? Sorry? Yes, why? Explain it. Yes. Yeah, so we have, as you see here, so we have 0.3 here and 0.8 here. So there's a 0.5 difference between these, so it's a 2 plus. And then here we have a 0.3. So that's the, yeah, so that's the reason. So this is and this is something that is used, of course, and in software. So now, as I said before, so protein identification. So what we do there is we take the mass to charge ratios and we try to go from there to determining what is a peptide, what peptide it is. Charges, you mean? Yes. I mean, if you have bigger in MALDI, I haven't seen that. In electrospray, definitely. Even if you have a small protein, you can get those kinds of charges. In MALDI, I mean, I don't think I've seen much more than 15, maybe, for quite a large protein. That's maybe you could get something bigger, but it's difficult to get such hard. So identification, so we can measure them. I mean, modern mass spectrometers can measure masses down to a few parts per million. So we can get very accurate mass measurements, both on the peptide and on its fragments. So we can, but we still don't know when we have a fragment mass spectrum if it's if the charge is on the N terminus for that peak or the C terminus. So we have always this ambiguity with if it's a B or a Y ion that we need to resolve. And we have the other problem is that we don't get fragmentation between each pair of amino acids in a peptide. So we have missing information also. And the best way to fill in this missing information, the easiest way is if you have a database. And that's what the first exercise is about, is to take, I don't know if you have downloaded the 2 files that have the extension MGF. If you can start searching those while I continue with some of the slides, so that you have the results. So the first part is to just take those 2 files and go to this link to Tandem and do the search. So only thing you need to, so you can use the default parameters, except for that you should change that it's Saccharomyces cerevisiae that you should search. So if you do those searches, we can then continue. And the task is to look at the list of proteins and try to then, we can break into smaller groups and then discuss so if you can explain why the different proteins showed up in this sample. So it's the sample is someone genomically tagged NAP1, which is a nuclear pore protein in yeast, and did an affinity purification. And they did two different conditions. One, they washed very carefully, so that in that sample we should see very few proteins. And then the other case, they didn't wash it very well, so that we should see a lot of proteins. And so, and we also will see some nuclear pore proteins. We will see some carioferins, which are easy to understand why they're there. But then we'll see other proteins that we should you'll have to think about why they are there. And then we'll come back to this later. But yeah, so did you all find the files? So while you do that, so I'm going to talk about how to do this in a more manual way. So first, since we have a fragment mass spectrum, now if we're going to interpret it, so there are two ways to interpret it. So now, the first we're going to assume that we know the sequence. And we're going to see if we can understand the spectrum and explain all the peaks. And so what we want to go from is that we want to be able to label all the peaks, whether they're Y or B ions, and between which amino acid the fragmentation happened. And we want to be able to annotate the sequence and show how much evidence we have for fragmentation between different amino acids. So the way we show it here, it's a small cartoon showing the length of these lines between the amino acids are proportional to the peaks in the spectrum. So this peak corresponds to this fragmentation, and this smaller peak corresponds to that one, and this large blue one is the red ones are the Y ions. So the charge is kept on the C terminus, and the blue ones are the B ions, where the charge is on the N terminus. So for this, the fragmentation between this isoleucine and leucine, we have a lot of evidence because we have both strong Y ions and strong B ions. So this is the whole point. And so the different strategies are either now, first we look at the spectrum and we have the sequence. So then we can calculate all the masses of the B ions. And we can calculate the masses of the Y ions. And then we can just match and look for these masses. So there, and we see that in this case, this is a very unusual case because we actually can see a lot of the B and Y ions, and we can explain the whole spectrum. But, so for example, if we look at this this fragmentation between this leucine and this phenylalanine, we see that the B ion that corresponds to that is 292. And the Y ion that corresponds to that is 875. And then we can take another step. We see that the difference between these two peaks is 113, and these two peaks is 113 corresponding to leucine. And then we can continue walking along the spectrum like this. And so what we see is that we get these differences between peaks that correspond to amino acids. So, but the general case is the opposite, that we don't. It's not that we have a sequence and a spectrum and we want to just confirm that they're matching, but what we take a spectrum and we have the list of amino acids and maybe some modifications also. And we want to deduce the sequence from that. So what we can do is one way is to take all the masses and make a big table for all the differences between all the peaks. So we end up with something like this. And we can then look at the ones. So these are the mass differences that correspond to amino acids that have been marked in green. And then we can switch to the amino acid symbol. And the case always, what we have is isoleucine and leucine have exactly the same mass. So we will usually won't be able to tell the difference. Sometimes if you do some quite esoteric higher energy collisions, you can get different fragmentation patterns, but almost no one does that because it's too complicated. So now what we do is we have a bunch of amino acids, but now we have to put them in order. So if we start here with at M over Z 260, we see that from 260 to 398 there is a glutamic acid that's between, corresponds to that. So then we can continue 398, so we jump down to 398 here, and the next one is an aspartic acid. So then we go to 504, and then we have another glutamic acid. And then we continue like that and walk down. And then finally we reach the end of one. But you see that there's still amino acids left. It's because we walked through the spectrum through one type of the ions. We still don't know whether it's the B ions we walked through. And then now we're going to walk through the other way. So then we start here. And in this case, there's an ambiguity. It could be a proline or an isoleucine, leucine. But we've already used this mass here, the 398, so it should be, that can't be. And we have the isoleucine, leucine. So then we walk through again, resolve ambiguities. And remember, this is a very good case. It's usually not this easy. And we can read off the sequence. So now we end up with this. So in one direction, we have this sequence, and the other direction we have this sequence. And luckily, it actually is the same, just switch. So that's good. But we still don't know the orientation. So what we can do is then we can look at the full peptide mass. We have the difference between this one ion, the 1079 here, which is at the end after this G. We see that to get to the full peptide mass from there, we have a serine. So now we know the direction, that this is the N terminus. And then we can confirm it and get the last amino acid from the mass. But here now we know that this is a Y ion, so we subtract 18. And so then we get that it's either a K, a lysine, or a glutamate at the end. So there is a difference between the lysine and the glutamate. But the mass difference is very small, and this spectrum was collected so that we can't tell that difference. So now this is what we end up with. We get the sequence, but we always have the ambiguities of isoleucine, leucine. But in this case, because we don't have very high accuracy, because we had the difference between this last ion, the 1079, and the full M plus H of the peptide was exactly 87. So if you because then that's how it works for the B ions, but for the Y ions there is you have to add water also, like this. So that's why we know that. So now the main, so this can be, for very good spectra, this can be done by hand, and even for not so good spectra, you can do it as long as all the information is there. But if you can imagine, if there is missing information, it gets very complicated. And that's, but you're going to also play with a few spectra afterwards at the end to find where they are phosphorylated. And it's the thing is that if you don't, and this is the case when you don't have a database, this is what you have to do. And there is some software out there that can do this or can help you with it, but it's really far from perfect. And it's still a problematic case when you don't have a good database. So, so then in addition to this, other problems, when we do fragmentation, we also have neutral losses. So we lose water, we lose NH3, which adds to the complexity. There are modifications. We have background peaks that we can't explain from the spectrum. And the worst thing is the incomplete information that we have. But database search is really good at filling in this incomplete information. It's really, because you have, if you calculate how many possibilities there are even for a small peptide, so you have, let's say, 10 amino acids, you have 20 possibilities at each position. How many possible peptides are there? Sorry? No. So you can have any amino acid at any position, and you have 10 positions. Sorry? 10 Sorry?
10 raised to 10.
Oops. Sorry. No, yeah, so almost 20 raised to 10. Yeah. So it's a lot of possibilities. It's an enormous amount of possibilities, even for such a short peptide. But only a very small subset of that is actually used. And especially if you know which species it is. If you, let's say, only search human proteins, it's only a few 100,000 peptides that are used out of this enormous space. So the data, that's why the database search is so much easier and much more automatable also. As you see, so have anyone gotten any results back yet? No? Yeah? Only some of you, or? Okay. Yeah, let's look at that a little bit later. So the database, what one does is that one mimics the experiment in the computer. So what we have here in the experiment, for example, we take a cell culture, lyse the cells, extract the proteins, digest them with trypsin, separate them with liquid chromatography, and fragment a lot of peptides. So then what we can do is, in the computer we have a sequence database. And of course, we should construct this sequence database so that it contains all the relevant sequences and nothing else. That would be ideal. Usually we don't know exactly what's, how to do that, but we, for example, take all the human proteins from the reference sequence. But we actually know that, I mean, every human has variants, so that some amino acids will be different. But usually we just ignore that. And in most cases, that's not a problem. So we take the sequence database, take the first protein, digest it according to the same rules, so trypsin cuts at arginine and lysine. Then we take the first peptide and calculate all the fragment masses and then compare what we measured to what we calculate. And then there are many, many different scoring algorithms for this. But the main thing is that we pick one scoring algorithm, but then we also test the significance of the result. So we repeat this for all the peptides and for all the proteins. And then we get a long ranked list for all the peptides for each MS-MS spectrum. And then we pick, then filter out, so we just pick the ones that are significant. And so the way that the X-tandem that you're using tests the significance is that it calculates an expectation value. So this is, you came across the expectation values with the BLAST, and so X-tandem does it in a similar way, that it assumes that the distribution of random hits is distributed according to an extreme value distribution, and then it can extrapolate to very large scores to test the significance of the high-scoring peptides. But if we take one step back and look at how much information is there in a single mass measurement. So now we're not talking about the fragment spectra, but just the mass of a single peptide. This here we have on the x-axis the tryptic peptide mass in Dalton and the number of matching peptides for different mass accuracies. And so, for example, if we go in here at 1 ppm and at a mass of 2000, there are about 4 peptides on average that match that. So just by measuring one peptide mass, we go from hundreds of thousands of proteins down to a single one in human. We can see the distribution here. So it's it ranges, but the average is 4. But as you see down here, we have the 0.1 ppm and 0.01 ppm almost overlapping. So there's really no improvement to be gained while to go to a lower mass accuracy. And can anyone tell me why? What happens there at that point? When we measure the mass, but we really can't determine, we don't get more accurate. So what we've reached is we can determine the elemental composition at that stage. So we have such high accuracy that we can tell exactly which atoms are there, so which amino acids. So the only thing we don't know is the order of them, and that we will never get from that measurement, of course. But that's why we, at that point, don't get any more improvement. And so if we do this for an organism with a smaller proteome, we get a much better accuracy. So at the same 1 ppm accuracy, tryptic peptide mass of 2000, most of the times it's just a single, defines a single peptide. Okay, so let's go back now to, so who has gotten some identifications?
I submitted it. I don't know if I chose the parameters correctly.
So you changed it to yeast.
Yes.
And did it come back?
It did.
Yeah. Okay, good. So I mean, you can use the default parameters. That should be fine.
I submitted the more stringent wash and got back a lot of things that I would think would associate with that.
Okay, like what kind of thing?
95, phosphorylated NUP1, F1, not phosphorylated, some human keratin, not surprising. Trypsin.
So where do you get human keratins from?
Hands.
Yes. But also, I mean, in general, old reagents. So it's good to make new reagents all the time. But it's almost impossible to avoid getting keratin in there. And trypsin there's some left of the trypsin that you added, of course.
ESA, ribosomal proteins.
Yes. So ribosomal proteins, of course, another thing that often shows up because there's a lot of them. But particularly in this case, it's even more probable because those are the ones that get transported most also through the nuclear pore.
F-170.
Yeah.
Enolase 2.
Yeah, enolase 2 is another high abundant protein. So did anyone look at the that was the more stringent wash, or yeah, the less stringent wash. Yeah. Okay.
There's similar proteins like NUC1.
Anything else that Dylan didn't mention?
The number of hits is higher than the wash.
Yes. So you get much more. Yeah.
129
Yeah.
versus
Yeah. And did anyone see sheep keratin? And so what is that from?
See what?
Sheep keratin.
wool.
Yes. Wool sweaters. That's so that one always sees a lot of also. So one, yeah. So it's a good idea to try to minimize these things, but definitely you'll always see those things. So then these we can look at this later. The other, the question B here, but so let me go back to. Oops. Right, how do I? So that was identification. So now quantitation is much more problematic in general than identification. And the reason for that is that the strength of the signal is very much dependent on the peptide sequence. So if you mix together two different peptides with different sequence, equal amount, depending on how you, what solvents you use, how you do the separation, you can get one peak being 10 times the size of the other, or the other being 10 times the size of the other. So you really need to compare the same peptide sequences, or have an internal standard that and which has also the same sequence but heavy isotope labeled. And this is why what people do mainly in proteomics is compare the same peptide or same protein in different samples, but not to determine stoichiometry, where you in the same sample have to compare different sequences. That is much harder. So in general, you have this experimental workflow. So what you're interested in is the concentration of one protein in the sample, or many proteins. But what you measure is the intensity of a peptide. And then you have all sorts of losses here that are dependent on what the protein structure is, and also on the sequence of the peptide. So if you know all these losses, of course, you can write an equation to calculate the intensity that you will measure of a peptide, how it depends on the concentration, but that's not the practical thing. So one thing we can do is, for example, what's called label-free quantitation, where you try to keep all the conditions as constant as possible and analyze one sample after the other and then compare them. Now, of course, it's not easy to do that, especially if you have many samples. Let's say you want to run 100 samples and it's going to take you 3 months to keep all the conditions, the columns in the same shape and not going bad, the temperature in the room, and so on, the humidity, all that matters. So, but if you can do it, and often people usually with label-free just do smaller experiments, a few samples that you can run during a day, maybe. What you can do is if you see a ratio, intensity ratio between two peptides that belong to each corresponding protein, then you can say that the ratio of the concentration of proteins is the same as the ratio of the peptide intensities. But it has to be the same peptide in different samples. And then what often happens, so let's say you have a protein where you have measured many different peptides. You're going to, of course, get slightly different ratios. And you're sometimes going to have outliers. And it's still a discussion of how to sort of combine these different measurements from the same protein in a good way. So sometimes you can explain certain thing why a peptide should be an outlier. Let's say it has a post-translational modification in some cases, so that that would, of course, could give rise to an outlier. But a lot of cases, people don't really know what's going on. So what you often, a simple solution is you take the median of all the peptide ratios. And that's so far it's still an ongoing discussion how to do it. So the other extreme now is when you do metabolic labeling. So you grow up you do a cell culture in the regular amino acids and one with heavy amino acids. So heavy meaning you add a few carbon 13s or nitrogen 15s, or you replace so go from carbon 12 to carbon 13, nitrogen 14 to nitrogen 15. So you get a few more masses on there. So then now the nice thing is you mix it together, and provided that the heavy amino acids don't affect the cell culture, which they probably do a little bit, but hopefully one can ignore that, then all the other steps, everything is together so that they should behave the same way. So now usually if one compares the metabolic labeling with the label-free, one gets a much better reproducibility. It's usually a big difference. So what you end up with in the spectrum here is that you have one light isotope distribution and one heavy. So because of the isotope distribution, it's good to at least have a distance of 4 Dalton, at least. Usually people use 6 to be able to separate the distributions properly. So now these are just 2 different ways, but then, as you can imagine, people have thought about introducing labels at every step in between here. So you can label the proteins, you can label the peptides, and so on. Then one variant of that is that you synthesize heavy peptides and you mix them in after the digestion. and get again, so as an internal control. But the problem here is, which a lot of people have seen, that it's asymmetrical. So here you digest your sample, but the peptides you mix in. So often you can get, one thing that you can add here is also a peptide antibody enrichment step. But what you can get is quite large systematic errors if your digestion doesn't work very well, or if your mixing doesn't work. So you can get synthetic, you can get quite large systematic errors in both directions. And people used to refer to this as absolute quantitation, because you can measure the how much synthetic peptide you made quite accurately. But the problem is that in the mixing and in the digestion, it really doesn't work very well as an absolute quantitation. But it works pretty well for as an internal standard. So now the question is, how then do we estimate peptide quality from? So we measure these peaks in the mass spectrum. So there are a few different ways. So usually you have a background. So in this case, that's what this red line indicates, that we want to subtract out the background, so that because otherwise our measurements are not good. But that's usually quite straightforward how to do that. But then we have these peaks, and so one way would be just to take simply the height of the peak. And then we can also decide on, do we just want to use the first peak in the distribution, or we want to use the 3 first ones, for example. That's some people use one, some people use the other variant. Another thing we can do is we can fit a curve, and if we know the shape. And usually we don't know the shape, and it takes too long to do that. And then another way is to do the peak area. So there are both advantages and disadvantages for each of these, and in general the advantage of the peak height is that it's resistant to interference. Let's say we have a peak and then there is a shoulder on it from another interference from another peak. If you just take the height, that will have be very little influence on what happens to the height. But the peak area, of course, will be much larger, because especially if we have noisy peaks, it's not easy to determine where it starts and ends always. Then the fourth way to do the quantitation is what's called spectrum counting. So here we sort of use the identification, and just count how many spectra we could identify peptides that belong to a protein. So we just count the number of spectra that corresponds to a protein. And this is very popular way to do it. It's not the most accurate way to do it at low intensities, but it's extremely resistant to interference, because we can identify much better than we can quantify. So we have quite reasonable statistical model to decide when it's a good identification, when it's not, but then probably the main reason why people are using it so much, it's so easy to implement. And actually, so that's what we're gonna talk. One of the exercise for the quantitation is actually on looking at spectrum counting data. So now, just to summarize, so I've talked a little bit about the structure of mass spectrometry data, and also about how to identify and how to quantify proteins. So next time, Stuart is going to talk about gene expression analysis, and actually what if you do protein identification and quantitation, then make a table of that has, let's say, proteins as rows and samples as columns. Then you can apply the same gene expression software as Stuart will talk about next time. So then the protein quantitation exercise is also on the website. So if you want to download those data sets also, so it's one text file and 3 Python scripts. And we're going to go back to that, but before that, and then there is also, oh yeah, so this is all, these descriptions, that's from the homework. So if you go to the homework, there is also a table that shows the fragment masses, the B and Y ions of a peptide. And it also has 2 spectra, one phosphorylated and one unphosphorylated. So that's what, if you can start working on these things now, and then Pam will, and I will be here also to help you with that.
I can start early. I'm just going to go through your Python.
Okay.
Because like this, I know is like very absorbing. So once you start, you're not going to want to stop. It's like eating a bag of chips.
Yeah.